I'm Skylar Litz, and I will be presenting on the project I've been working on this summer as a trainee with the Mayan Lab. My project was to create appyters that produce unique visualizations, both static and dynamic, of enrichment analysis data. I want to start by giving a background on Enrichr and enrichment analysis. Enrichr is a web-based application for enrichment analysis that was first developed by the Mayan Lab in 2013. Since its creation, it has been used to analyze over 26 million gene sets, has had almost 250,000 unique users, and has been cited over 4,000 times. In order to understand the appyters I've created, it is essential to understand what enrichment analysis is, why it is useful, and how it is performed. Enrichment analysis allows biologists to understand the underlying biological functions and processes of a set of genes by comparing the set of genes with a library of annotated gene lists. Experiments involving transcriptomics or proteomics often yield large gene sets which can be analyzed using enrichment analysis. There are different ways to compute enrichment analysis, but the most common method, and the one used throughout this project and in Enrichr, uses the Fisher exact test. This method takes a user-inputted gene list and it compares it to libraries which are made up of many gene lists, each of which has a term associated with it. A p-value is calculated for each annotated term in the library. This is done by comparing the observed frequency of an annotated term with the frequency expected by chance. Annotated terms with a p-value below a certain threshold, meaning that they are overrepresented in the inputted gene list, are determined to be enriched. The example shown here is actually using a real gene set and a real gene set library from Enrichr. The gene set used in this example and throughout the presentation is an upregulated COVID-19 signature from the human lung, which was extracted by my colleague Jesse. We will follow this gene set through all the Appyter demonstrations and explore how the visualizations are useful for analyzing the set. Now, Enrichr and enrichment analysis in general are very powerful tools, but it's easy to get overwhelmed by the large amount of data that's calculated after performing enrichment analysis. Each of these tables represents one library. Enrichr has over 150 libraries available, and libraries often have thousands of individual terms within them. Enrichr actually also calculates even more than just p-value, so there are actually even more columns in each of these tables. You end up with lots and lots of data and have to find a way to extract useful information from it. Here's a look at the visualizations that Enrichr currently supports. There's a dynamic bar graph and a clustergram. Then for some libraries, a network and a grid visualization are also available. As mentioned before, Enrichr has been cited over 4,000 times by different publications. There is a need for software that can create publication-ready visualizations of enrichment analysis data. This summer, I have been able to develop 3 new visualization methods, each utilizing the Mayan Lab's Appyter framework, which will allow scientists to generate publication-ready figures quickly and easily. First, we have the Canvas Appyter. For some background on the Canvas Appyter, the Mayan Lab developed a web application called Network to Canvas in 2013. This was developed as a way to visualize large networks on a square canvas where each square represents one node. The nodes are clustered next to other nodes that they are most connected to. Then, on this canvas, enrichment analysis results can be displayed by brightly coloring nodes that are highly enriched with an input gene set. Here's an example of Canvas visualizations that were published in a paper in Nature about genes disrupted in autism. You can see that the canvas provides a compact way to visualize the enrichment analysis results, but these canvases took a long time to prepare and squares are not the best tessellation option. Gate is another piece of software worth mentioning. It was developed by Alex Lockman in the Mayan Lab. In the visualization here, each hexagon represents one gene. The figure on the right is from another paper published in Nature which utilized this approach. The idea with the Canvas Appyter is that we can leverage the tessellation of hexagons, but instead of each hexagon representing a gene, each hexagon represents a gene set. Here's a look at the input form for the Canvas Appyter. Users can input a gene list and select 1 or 2 Enrichr libraries. There are also additional options including color choice, whether the top 10 or the top 20 most similar hexagons should be colored, as well as the option to compare results from 2 libraries and separate canvases output at the same time. Here's a sample output from the Canvas Appyter visualizing the COVID-19 signature on the KEGG 2019 library's hexagonal canvas. We will return to this example later, and I will provide information about the clusters and what gene sets they represent. In general, each hexagon on the canvas represents a gene set from the selected Enrichr library. The hexagons are colored based on Jacquard similarity index between a user-inputted gene list and each annotated gene set in the library. The colored hexagons represent the gene sets from the library that are most similar to the inputted gene list. In general, the more similar a gene set is compared to your input gene set, the darker its hexagon appears. If you hover over a hexagon in the output figure, the gene set name and similarity index is displayed. You can see on the canvas here that the colored hexagons are clustered together, just like in Network to Canvas. This is because the Enrichr libraries were pre-processed using a simple simulated annealing algorithm, which I will describe now. For the algorithm, first, the overall fitness of the canvas is calculated. The fitness of the canvas is measured by first summing the similarity indices for all of the neighbors of each hexagon, as shown here. Then, all of the totals for each hexagon are summed. This gives us a quantitative value for the fitness of the canvas. Next, two hexagons are chosen at random and swapped with each other. The fitness of the canvas is recalculated again after the swap. If the fitness increases, the swap is kept, but if it decreases, then the swap is reversed. In the example here, the swap increases the overall fitness from 31.7 to 31.9, so it would be kept. Because the fitness function and algorithm seek to maximize the similarity of each hexagon to its neighbors, the hexagons end up clustered with hexagons that are most similar to them. And remember that in this canvas, each hexagon represents a gene set. So the hexagons close to each other have overlapping gene sets. Here's the example of the COVID-19 gene set compared to the KEGG library again. I've labeled the colored hexagons with their name and associated p-value. You can see that the clusters make visualization of large amounts of enrichment analysis data compact and easy to look at and analyze. The next Appyter I created produces Manhattan plot visualizations. Typically, Manhattan plots are used to determine variance. In the figure here, you can see that the x-axis shows location of genes on a chromosome, and the y-axis shows the negative log of the p-value for each gene. Single nucleotide polymorphisms can be located easily because the areas have much lower p-values than the rest of the genes, so when the negative log of the p-value is plotted like this, they stand out clearly. In the Appyter I created, rather than being grouped by chromosome on the x-axis, gene sets are grouped by library. So while the plots are not useful for finding variance, they are useful for visualizing large amounts of enrichment analysis data at once. Here's the input form for the Manhattan Plot Appyter. Users can input a gene list, select Enrichr libraries, set a value for a significance line, select output options for saving plots, and more. Once everything is input and submitted, the Appyter creates two output plots. The first output is a static Manhattan plot. Pictured here is a sample output using the COVID-19 upregulated signature. The y-axis is the negative log of the p-value for each term, and the x-axis consists of terms which are grouped by Enrichr library. In the static plot, which is shown here, if a significant value is provided, then the significant terms are labeled and their p-values are displayed in a legend to the side. The Appyter also outputs the dynamic plot, which can be seen here. The dynamic plot has the same layout as the static plot, but allows users to hover over each point to see the associated p-value and term, like this. Overall, the Manhattan plot provides a useful way to compare enrichment analysis results from multiple libraries at once, and the outputs are intuitive to interpret. The most enriched terms are the ones that stand out above the rest. Finally, we have the Scatter Plot Appyter. For some background on the Scatter Plot Appyter, we start with a tool that the MyOnLab created called L1000 FWD. This is a web application that provides interactive visualizations of drug and small molecule-induced gene expression signatures. The plot seen here was created by first creating a network, then using a clustering algorithm to create a scatter plot where points are clustered by similarity. L1000 FWD was actually used very recently to successfully discover drugs that can trigger an immune response in the presence of COVID-19. For the Scatter Plot Appyter, clustered scatter plots similar to L1000 FWD were created, but using Enrichr libraries instead of drug and small molecule data. Here's the input form for the Scatter Plot Appyter. The user selects between 1 and 9 libraries from Enrichr and uploads a gene list. After submitting, here's a sample output. A scatter plot, like the one shown here, is displayed for each library where the significantly enriched terms appear blue. Each point on the scatter plot represents one gene set from the library, and users can display the associated annotation and p-value by hovering over each point. We will return to this output in a bit, and I will provide some more details for the results pictured, but first I want to explain how the scatter plots were created. The libraries from Enrichr were pre-processed using machine learning techniques to create clusters of similar gene sets. To do this, First, TF-IDF, which stands for Term Frequency Inverse Document Frequency, was applied to the gene sets to determine weights between 0 and 1 for each of the genes. If a gene is very common and appears in many gene sets, it is not very unique, and thus it is given a low weight. Genes that are more unique and would be more helpful in distinguishing one gene set from another are given a higher weight. Note that for any one given term, no genes in the gene list are repeated, so really only the inverse document frequency part of the algorithm is being utilized, but this still provides us the results that we want. Then, we have a big matrix, something similar to the one on the right. We use NMF, or non-negative matrix factorization, to split the matrix up into two matrices with non-negative entries that multiply to create this big matrix. This step is performed because in practice we have found that it has led to clearer clustering in the final scatter plot. Finally, UMAP, which stands for Uniform Manifold Approximation and Projection, is applied to one of the matrices resulting from NMF. UMAP is a dimensionality reduction technique, so it reduces the dimension of the adjacency matrix down to 2, so the results can be plotted and visualized on a two-dimensional scatter plot. Here's an example using the CHIA 2016 library from Enrichr. As a reminder, each of the points represents an annotated gene set from the original library. The different colors here indicate each of the components of the NMF model, so you can see how the NMF step helps to cluster the gene sets. The preprocessed libraries are stored in a public GitHub repository. Then, when the Appyter is run, they are loaded and enrichment analysis is computed, comparing the inputted gene list against the gene sets in the library. You can see that the enriched terms also cluster together, which makes sense given that similar gene sets are clustered together before enrichment is calculated. The plots shown here are again visualizing the COVID-19 up signature. Some of the most significant terms are indicated on the side. While it is difficult to label all of the significant points in this format, in the output plot itself, the hover feature allows for all terms and their p-values to be explored. All three of these Appyters are available on the Appyter catalog now, so anyone can create their own Manhattan plots, canvases, or scatter plots. Also available now in the Appyter catalog is the Enrichment Analysis Visualization Appyter, which will create each of these three visualizations, plus a static bar chart for one inputted gene set and one selected library at a time. That Appyter also outputs a downloadable table of significant p-values and includes a link to Enrichr so you can explore your full results. These Appyters provide publication-ready plots and figures so that scientists can quickly investigate and publish their enrichment analysis results in unique ways. I want to thank my collaborators, Daniel, Clark, and Megan, for always answering my many questions. I want to thank Sherry for her incredible organization of everything and for being such a positive, encouraging person throughout the program. I want to thank all of the lab members for their support and suggestions throughout my project. Charles developed the title field, which I used for all of my Appyters, and Jesse provided me the COVID-19 signature, which I visualized throughout this presentation, so special thanks to them. Finally, I want to thank Avi for all of his guidance and support throughout the summer. It's been an incredible learning experience, and I feel as though I have grown as a scientist and researcher, so thank you.